Good morning, good afternoon, good evening, wherever you are. My name is Glenn Pierce, and we're going to start the webinar on progress toward a cure for hemophilia, demystifying gene editing. I want to remind you that at the bottom of your screen, you've got a Q&A box, so please submit questions along the way, and we'll cover them toward the end. And then there is closed captioning available in English as well. I'm going to start with an introduction to genetic technologies, and I'm going to talk about three genetic technologies: AAV gene therapy, which we've heard a lot about, lentiviral gene therapy, which we haven't heard too much about, and gene editing, a newer form of gene therapy that I'm going to focus on. And then we'll take questions. Before we start, these are my disclosures. I work with a number of gene therapy and gene editing companies, and I have worked in both the hemophilia as well as non-hemophilia spaces. So let's start with the basics. What is DNA and what are genes? We have about 30 trillion cells in our body. So that's 30 followed by 12 zeros. That's a lot of cells. And those cells each contain DNA, and the DNA is the instructions for the genes that make us who we are. We have about 20,000 genes in our body, and when those genes express proteins, then they make us who we are. They make our constitution, what we look like, how we behave, how our metabolic processes work, and everything else. So that's where we start with the DNA. Now, the genes for hemophilia, there are two of them for factor VIII and factor IX deficiencies, hemophilia A and hemophilia B. And people without hemophilia, the genes are on one of the chromosomes that contain genes in the body, and it's designed to make functional factor VIII, factor IX proteins. If one has hemophilia, then there's a defect in either the factor VIII or the factor IX gene, and it results in either absent factor VIII or factor IX protein or a protein that does not work very well. Hemophilia A and B, they're rare, they're inherited, and they individually can cause a very similar kind of bleeding, diathesis. So individuals with hemophilia don't make enough of the protein, and that will lead to the inability to form good clots. So what's the goal of genetic technologies as it's evolved over the past 30 years or so? It's really to provide a new copy, a working copy of the dysfunctional gene so that the body can make its own clotting factor proteins. And that's as opposed to repeated injections of either factor VIII or factor IX clotting factors or other clotting agents, such as the non-factor therapies, the factor VIII mimetic or rebalancing agents. And so those carry a burden of therapy with them. They require repeat dosing. Some of them, such as the clotting factors, will have peaks and troughs. But the gene therapy will offer a constant amount of protein in the body. So there's multiple types, as I mentioned at the beginning, multiple types of genetic technologies that are being developed for treating hemophilia as well as many other diseases. The AAV gene therapy, two of these products are already approved. The lentiviral gene therapy, which I'll tell you a little more about, very exciting data, and gene editing using CRISPR. And so you may have heard about CRISPR in the popular press. It's a type of a gene therapy that really involves editing, which I'll describe in a few minutes. With each of these technologies, there's key differences. There are different benefits, different risks, and different methods for creating these drugs. What do all of these technologies have in common? Well, they target non-reproductive cells, either in the bone marrow or in the liver, and so the effects cannot be passed down to children. And once the therapy has been delivered, this is really a once and done process. Process cannot be reversed or stopped. Once the gene therapy is there, it's there to stay for the rest of that person's life. And that's whether it works or not. So in clinical trials, some of the clinical trials, the gene therapy hasn't worked, but it's still with that individual, with that participant for the rest of their life. So that's an important distinction from all of the protein replacement therapies that are used, such as the clotting factors or the non-factor therapies. So let's look at how these genetic technologies change DNA. If you think of your DNA as a book of everything you are, where every sentence in the book is a gene, all three of these technologies result in the addition of a complete sentence that can be read and understood. With AAV gene therapy, it's like adding a sticky note containing a new sentence on the outside of the book. That new sentence then is stuck there and can produce the protein for the gene. With lentiviral gene therapy, it's like randomly inserting a new sentence anywhere into the book. And that sentence can go into the middle of an existing sentence, for instance. And then with gene editing, it's like correcting small grammatical errors in a sentence or putting in a new grammatically correct clause into the middle of an existing sentence. So that's a good way to distinguish between the three different types of gene therapy that I'm talking about, as well as to demonstrate their differences. So how do these genetic technologies work? Well, they all use a therapeutic transgene. And the therapeutic transgene contains the instructions for where in the body the functional clotting factor protein should be made. So the therapeutic transgene is the factor 8 or the factor 9 gene. And then it is placed into what's called a cassette that also includes the instructions for that gene. So when should that gene get turned on? How much of that gene should get made into protein? That sort of thing. Now, these genes are transported by viral vectors. Why do they get transported into the body by a virus such as AAV or lentivirus? Well, because viruses for a 500 million years have attempted to get into cells of vertebrate animals and have perfected it. And so it's an easy way to get genes that we might want to put into the body into cells. And that's been established over the past 40, 50 years now, the ability to use viruses to deliver genes. Now, these viruses are made up of an outer shell that's protein that has been modified. And the inside of the virus, the part of the virus that can cause disease, such as influenza or HIV, for instance, all of that has been removed. And so we're only dealing with the shell, and we put our therapeutic gene inside of that cell. So now let's talk about AAV gene therapy as we move toward our goal of getting to differentiate what gene editing is. So with AAV gene therapy, we're delivering a therapeutic copy of factor 8 or factor 9 to the liver, and then it allows the liver cells, which are called hepatocytes, to produce the functional proteins that are released then into the bloodstream. So you can see on the left, the AAV gene therapy is administered into a vein through a single one-time infusion. And then once it's in the body, it circulates and it delivers the genes uh, in the vector to the liver, and then the body can begin, if assuming this works, to begin to make its own factor VIII or factor IX protein. So we have three gene therapies that have been approved in this country, in the United States, as well as in a number of other countries. And then one gene therapy has been approved now only in China. So we have Roctavian, valoctocogene roxaparvovec, for hemophilia A, treatment of adults. Uh, and it uses an AAV5 serotype vector. So there's a lot of different AAVs, just as there's a lot of different strains of dogs in the world. We have a lot of different AAV viruses that can be used that are all slightly different from one another. Uh, Roctavian is available now in the US, Germany, and Italy. And then there's Hemgenix, etranacogene dezaparvovec, and that's for adults with hemophilia B. That's available now in the US, the European Union, United Kingdom, Switzerland, and Australia. And then there's Becfis, fidenacogene elaparvovec, and that has been withdrawn from the market because it had poor sales. And then finally, available in China, developed more recently, is BBM H901, dalnacogene panparvovec. And that is very similar to Hemgenix for the treatment of adults with severe hemophilia B. So how does the AAV gene therapy affect the factor level? Well, there's no way to predict exactly what will happen when it's administered to each individual. But the individual factor levels respond, and it's also not totally clear how long the effects will last, particularly for the factor VIII gene therapy. And as I mentioned before, it's there forever. So it also cannot be redosed. We can't top a patient off, for instance, and give them a higher amount. And it's not able to be used in children at this point. So with hemophilia A, the levels will increase, go into the mild to normal range, but then decrease within the first year. And they may continue to decrease with time. Or in some patients, they may stabilize. But it's not predictable, and the durability has been an issue for the hemophilia A gene therapy. For hemophilia B, the levels increase, they go into generally the high mild range, and they appear to be sustained for a long period of time. A recent publication a month or two ago came out from one of the original groups, University College London and St. Jude's, that began doing work with factor IX gene therapy back in 2009 or so. And they treated those patients, and they've got data out 13 years demonstrating it's stable. And the Hemgenix that has been approved is using essentially the same kind of a cassette. And so we have every reason to believe that it looks like it's stable as well. There are some people that get a low response, and also some that don't respond to gene therapy. And usually that's understood, but sometimes, as in hemophilia A, it's not understood. So here's the 5-year data now, the most recent data for Roctavian, valoctocogene roxaparvovec. And what you can see is a big rise at the beginning within the first year, and then it begins coming down. But it doesn't stabilize. It continues to trend downward over this period of 5 years. It may be stabilizing here at a low level, but by week 260, we've gone from levels that are 40, 50% down to levels that have a median of about 6%. And so that's quite a drop. Now, despite that drop, one of the main outputs from gene therapy or outcomes is the annualized bleeding rate. And so what you can see here is an annualized bleeding rate of under 1 for the most part. And even at year 5, when the level has dropped quite a bit, uh, most individuals are not bleeding. It's annualized bleeding rate of zero point 0.6. So it has remained effective in the majority of patients for about 5 years, although some patients have had to go back onto prophylaxis. And then the 5-year data for etranacogene dezaparvovec, Hemgenix, is shown on this slide. And what we can see is that it rises during the first year, and then it really stabilizes. And it's stable, it's somewhere close to 40%, 35 to 40% over a period of 5 years. And what we can see here is that what you would expect with an average of 40% is an annualized bleeding rate that is fairly low. 1.33 in the first year, but then it drops down to under 1, and by years 4 and 5 is at 0.4. So it's proven to be very effective. And from this trial, only one person has returned to prophylaxis because they had a level of, I think, somewhere between 3 and 5%. So there are safety considerations with the gene therapy. Some integration does occur. Integration in 1 to 1,000 to 1 to 100 hepatocytes has been shown in a number of animal studies for this gene as well for these genes as well as for other genes. And while that doesn't seem like much, we have 140 billion hepatocytes. So in 1 to 1,000, that would be 140 million integrations into a person's liver. That means that there is a theoretical risk that this integration could trigger an oncogenic or cancer-causing event that has not occurred in humans at this point. What has occurred is liver toxicity, and so ALT elevations have occurred. ALT is an enzyme in the liver, aminotransferase. Elevations have occurred. And so in hemophilia A, in that trial, about 80% of patients had elevations in the first year. And then you can see from the graph that those elevations have continued in years 2, 3, and 4 in a minority of people. They're small elevations. They're not clinically important, but it does demonstrate that there is some insult to the liver that is present in the patients who have elevations. And then in hemophilia B, it's occurred in fewer numbers of patients, and it's really disappeared after the first year. So I want to move now to lentiviral gene therapy. We haven't talked much about that because there hasn't been much to talk about. But it has been used now in hemophilia A. It's also been approved in 2 other rare blood diseases, sickle cell anemia and beta thalassemia, another type of anemia. And so there is good experience working with it. And we have 2 groups that have been working with it and developing it for hemophilia, both for actually all for hemophilia A. And so what happens with lentivirus is that it's a cell-based therapy. And the person's blood stem cells are collected and then they're modified outside of the body. And I'll show you that here. So what happens is that the stem cells are collected just from peripheral vein. The blood can be collected and the stem cells can be purified out from the blood. And then they're incubated with a lentiviral vector. And this lentiviral vector integrates its genes directly into the body, into the chromosomes of the body, and the cell. And then once it's integrated, the cells are growing up into a larger amount, and then those modified stem cells are put back into the body, and they differentiate into blood cells that then produce functional factor VIII protein. And so by doing that, we can get a integrated form of the factor VIII gene to secrete factor VIII protein. There are some important safety considerations with lentiviral gene therapy. As I mentioned, it's like randomly inserting a new sentence anywhere in the book, even into the middle of an existing sentence. Inserting into the middle of an existing sentence or an existing gene does offer a theoretical risk of cancer. And so while that has not occurred with these current lentiviral gene therapies, it did occur with earlier versions of these gene therapies, but steps have been taken to correct for that and prevent the likelihood of that happening. Nonetheless, there are unanswered questions regarding safety of this therapy. So we have two ongoing clinical studies. One is at the Christian Medical College in Vellore, India, and the other is at the Medical College of Wisconsin. And I will speak about the one that is in India, since they had a full study published about a year ago. And so with this lentivirus, which has this name of CD68ET3LV for lentivirus, the lentiviral vector contains the factor VIII transgene that is put in to hematopoietic stem cells and progenitor cells that have been taken out of the body. So in culture, it's put in, and then they're put back into the body, and they differentiate into monocytes and macrophages, which are two cells of the immune system that are responsible for fighting infections. But these cells also have been changed to make the factor VIII protein. This is the cassette, so it's very similar to what you saw for AAV. It's got the transgene in it. It's got the instructions to make the transgene as well, which is also called a promoter. So what did Srivastava and colleagues find when they put this into 5 patients? Well, using an earlier production process, they got levels in the 3, 4, 5% range, which was very good considering this was the first time this was done. And those levels lasted through at least 2 years and are still being studied. But then in the last 3 patients, they modified the production process and they got levels of 30%, 70%, and 50% in these 3 patients. So high mild to normal range for these 3 individuals. And essentially, assuming this lasts, curative therapies. These individuals shouldn't have to worry about bleeding at this point. So stay tuned for more on that. It's potentially an important therapy, especially for individuals that don't have access to all of the therapies that we have access to, for instance, in North America and Europe. And this study was conducted in India, where patients don't have access to all of the most advanced therapies. So let's move on now to gene editing. And gene editing can correct small genetic errors in a gene, or it can insert new genes into a specific safe location in the genome. So instead of random locations like lentivirus, this can put the gene exactly where you want it, which is a safer kind of an approach. In hemophilia, the current technologies that are in development go into these safe locations. They're called safe harbors to incorporate the therapeutic copy of the factor VIII or factor IX gene directly into the cellular DNA. These are still in clinical trials. They're early trials. There are still many unanswered questions from this type of a therapy. But what we have now is we have for hemophilia A, still in preclinical, but moving toward the clinic, a compound called MGX-001. that is for hemophilia A, and we have two that are in the clinic now for hemophilia B. One from Regeneron and one from B Biopharma. They're both in early phase 1, 2 studies and remain under clinical investigation. No data have been released yet on these therapies. So what is the gene editing machinery? Well, it's collectively known as CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats. But let's call it CRISPR. And it alters specific sections of DNA in specific cell types. This is the machinery, and it's comprised of two things. One is a Cas protein that is an enzyme that can cut the DNA. So it's like scissors, it cuts the DNA. And then there's a guide molecule that is attached to it that guides this scissor-like protein enzyme directly to a spot that you want to cut in the DNA. So remember, I said we've got 20,000 genes, we've got a lot of DNA. This guide molecule takes it precisely to one particular spot within those 20,000 genes. And so it does that by using a dual infusion in vivo with our old friend AAV and then the editing machinery. They're both infused at slightly different times. The AAV contains our transgene, factor VIII, and the editing machinery contains the cutting enzyme and the guide RNA. And so doing that, it's infused into the patient, and then this makes its way to the liver because the AAV tends to go to the liver, and that's where the effects occur. And then this other approach is called autologous cell transfer. And this is done outside of the body. So blood cells are taken from the body. They're incubated with the AAV vector in culture. And the vector then puts the gene in along with the guide RNA and the scissors to cut into a specific spot in the cells that are in culture. And then those modified cells are then put back into the body. And that's the approach that B-Bio is taking. So delivery via the dual infusions from Metagenomi. The therapeutic gene is delivered via the AAV, as I mentioned, and the gene editing machinery is delivered via lipid nanoparticles, which are also called LNPs. And these are just bags of essentially fat that contain the scissors-like protein and the guide RNA. And they are protected when they're delivered into the circulation, and they also home into the liver. And then the approach with the autologous cell transfer, as I mentioned, the therapeutic genes delivered via the AAV, and the gene editing machinery is really delivered the same way via these lipid nanoparticles. But in this case, they are directly infused into the body and home into sorry, they're not delivered into the body. These are cultured. These cells are cultured, and they're delivered into the cells that are in culture. And then the cells are delivered back into the body, which have been modified. So what happens exactly? Well, the scissors protein cuts the DNA in a particular spot that has been guided by the instructions or the guide RNA. This guide molecule then takes it right there, gets cut, and the new gene is inserted. We all have natural DNA repair processes that will repair our DNA and allow the new gene then to be inserted. So gene editing, as I mentioned at the beginning, it's like correcting small grammatical errors in a sentence. And in this case, we insert a new grammatically correct clause into our DNA in the middle of an existing sentence. So I'll just show you a little bit of data from preclinical work in non-human primates. This is the Regeneron factor 9. And what you can see here, all of these lines represent individual primates. And you can see the amount of factor 9 activity, normal being 100%. So they were able to achieve at least 100%, and in some cases, much higher amounts, close to 1,000% factor 9 in this dose response experiment. And so you can see the two doses. The one dose is getting us to an average of about 110, 120%. And the second dose is getting us to an average of 400%. And so the company has gone in with a smaller dose to determine if this will work in humans. For metagenomic with factor 8, the factor 8 activity in non-human primates, they did a lot of different groups. They mixed and matched the amount of AAV and the amount of lipid nanoparticle. And what they found, if we just look here, the constant amount of AAV, they found with increasing amounts of lipid nanoparticle, factor 8 activity going from 15% to 50% to about 70%. And so this is the basis for which they will develop their clinical trial for patients, perhaps starting next year, late next year. And so coming back to the factor 9 and the ex vivo approach that I talked about, the AAV and the guide RNA with the scissors are here. They contain, the AAV contains the factor 9 Padua mutation, which gives a higher activity factor 9. The blood is isolated from the body. B cells or B lymphocytes, which are part of our immune system that make antibody, they've been isolated and they've been engineered to put in the factor 9 gene. Then they're expanded and differentiated into a cell type called plasma cells, which is the cell type that makes antibodies. But in addition to making antibodies, this cell type also can secrete the Padua factor 9. That is then infused back into the body. It goes into the bone marrow, which is where these cells normally reside, and should make factor 9 protein that would be released directly into the circulation. So what about gene editing safety? Well, as I mentioned, all the gene editing we're using is using AAV. So all of the issues with safety in AAV remain for gene editing. We can have some acute toxicity in the liver. We can have some subacute toxicity that goes out over a period of a year. And then we have this potential, theoretical potential for long-term toxicity. Although, as I mentioned, no oncogenic events, no cancer-causing events have been seen to date. And then what about whether gene editing can mitigate any of the toxicity? Well, the factor 8 gene is going to be easier to deliver than in direct AAV gene therapy because it winds up being smaller. It doesn't have all the instructions with it, and so it's easier for the AAV to deliver it. We also are continuing to see improvements in AAV manufacturing, which has been an issue in the past. Detecting the ability to make high quality AAV. So that's continuing with this next generation of therapies. And then the goal really is to eliminate AAV so that we don't have to deal with the virus at all. But that is not yet been possible to take a lipid nanoparticle, put the factor 8 or factor 9 gene inside of that, and get a sufficient amount of it into the cell in order to be able to get protein made. So we don't have that ability yet, but people are working hard to try to construct lipid nanoparticles that could replace AAV in the future. Lipid nanoparticles, however, have their own safety issues. And these, as I mentioned, are bags of lipids that can cause inflammation. They do home into the liver. They can cause some dose-dependent inflammation within the liver. Um, but as you also will perhaps recognize, these LNPs have been used, for instance, for the COVID vaccine for years now, and they really have minimal toxicity when they're injected into the muscle. So it's really just in the liver where they've got some toxicity. And so the patients do need to be carefully monitored for toxicity and given steroids if required. So what do we know about safety? Well, both AAV and the lipid nanoparticle can cause some liver toxicity. The guide RNA that directs it to the site goes directly to the target sequence, but it could potentially have off-target effects. So it could wind up taking your scissors somewhere else in your genes and disrupt another gene. There are a lot of tests that are done to look for that and make sure that that doesn't happen with the particular design that has been made. But it is important to recognize that this is still early technology, a work in progress, and requires continued improvement, continual improvement. And so, as we think about these early clinical trials, we're back to the concept of shared and informed decision making, discussions between people with hemophilia and their healthcare team, their families, on the risks and the benefits of the therapies. So at this point, you might be wondering, do we need gene therapy? We've got lots of other therapies available. But what gene therapy does is really relieve the burden and provide, once we know that it works, of course, provide for the concept of a hemophilia-free mind, which Cedric Hermans and I have talked about over these last several years. It enables the permanent insertion of a gene into a specific site in our genome. That should provide lifelong production of the new protein because as cells divide, that gene goes along with those cells and works in the progeny cells. If it's shown to be safe, this can also treat newborns and children. And it aims to provide a sufficient amount of factor VIII and factor IX to get us to zero bleeds and that hemophilia-free mind. And importantly, this can provide a proof of concept for other bleeding disorders. So that includes von Willebrand disease and rare bleeding disorders, factor VII, factor X. Once we demonstrate whether this can work effectively and efficiently with factor VIII and factor IX, then it's a matter of working with the FDA to determine an expedient path or a very short path to be able to work in more rare bleeding disorders. So the answer to we need gene editing, the answer is yes. Not only for our bleeding disorder genes, but actually for many other monogenic disorders as well. So I want to call your attention to a gene editing booklet that we have produced at the World Federation of Hemophilia. It's available on the e-learning platform. You can shoot a picture of the QR code, which I think will go into the chat as well. It will be getting translated as well. Plus, slides and a recording of this presentation will also be up very shortly within the e-learning platform. I want to thank Regeneron for supporting this initiative, and a big thank you to my two colleagues at the World Federation of Hemophilia, Gwen Kaiser and Mas Nakachi, for the development, working with me on the development of these slides, as well as this booklet. With that, I'll thank you, and we will move to questions and answers. And now I have to figure out how to get to the questions and answers. Here we go. So when will gene therapy for treatment in terms of cost of hemophilia be? So the treatments, the gene therapy treatments are very expensive when we look in, for instance, North America, Europe. Uh, um, but amazingly enough, because of the high costs of factor treatments, for instance, the high cost of factor nine, they're cost effective. Factor nine costs an average of around $700,000 a year for an adult in the United States. And gene therapy at 2.5, 3.5 million is cost effective because assuming it lasts for at least 7 years. And I think it's pretty clear that it does. So while these are very high costs, we can expect them to come down as prices are get negotiated in the real world. And they certainly have come down for Roctavian, the factor eight gene therapy. They do provide for multi-year multi-year treatment. And then a question here from Brian. What are the potential implications, if any, for hemophilia gene editing and the recent FDA decision to place two Intellia CRISPR trials on hold? And so what Brian is referring to is a different gene therapy that uses a lipid nanoparticle very similar to the lipid nanoparticles used by the Regeneron gene editing trial that did cause a patient death. And the patient got a huge inflammation in his liver. We don't know all the details. We don't know what other illnesses that patient had. We do know that he was 80 years old, but we don't know about his other diseases. So I don't know that it has any long-term effect at this point, Brian, but it is something that we are going to need to watch very carefully. And this is all part of the unintended consequences of new technologies, which is why they're not approved by any stretch of the imagination for hemophilia at this point, but they are undergoing early clinical trials with careful monitoring. Is there any chance of gene editing could be reversed if complications arise? Um, no, the answer is no. This is a once and done therapy. It's integrated into your genes. And for that reason, it cannot be removed. Hassan, hi. Thank you for such a detailed update. You mentioned for Roctavian, there's a reduction in bleeds despite lower values. Well, the reduction in bleeds comes from the fact that the patients still have some of the transgene factor eight on board, and so as they come down uh, in factor eight activity, they still are protected. And so even though they're at 6% on average, some of those patients have gone on prophylaxis, but for the patients who remain, they've got good protection at that level. So that's essentially an average of that a patient with mild hemophilia would have, and patients with mild hemophilia don't bleed very often. Also, have we been able to develop consensus on a purchasing model, and what impact would outcomes-based agreements have? Well, so there are individual purchasing models for both the factor eight gene therapy and the factor nine gene therapy. Uh, it's with individual governments, and sometimes it's with individual institutions. So it is a cumbersome, uh, uh, difficult process to go through to get approval to use this approved product, one of these approved products, in your hospital, your institution. But the companies are working through it with the institutions, they're working through it with governments. It's been slow, but we will continue to see more and more countries and more institutions, particularly for factor nine, in terms of the countries, um, uh, make it available. Does the hemophilia mutation predict response to gene therapy? Um, no, the answer is probably no. We have mutations where we where we have protein that doesn't work, that still circulates, and then we have mutations where no protein gets made. But as we can see from giving the factor products, that doesn't have an effect on whether or not a patient responds to those factor products. So it really wouldn't have an effect on the gene therapy. Roshni, hi. Can you explain platelet delivery for gene therapy? Well, that's the other lentivirus therapy that I didn't have time to cover, but I mentioned it. Um, and what's done there is that the lentivirus is used similar to what Srivastava and colleagues did with monocytes and macrophages to make the factor VIII, except this time the instructions allow it to be made only in the platelets. And so as the platelets circulate, when they get to the site of a wound, they basically explode and release all of their contents, and that would include some factor VIII at the site of a wound, and that factor VIII then would be able to provide for hemostasis, provide for clotting. And so that is an early clinical trial. They have tested a few patients, and with the first patient, they published a letter to the editor in the New England Journal earlier this year, and they demonstrated that this patient didn't bleed for about a year and counting. So looks like that it may be an effective approach as well. Any gene editing for hemophiliacs with inhibitors. So the platelet gene therapy, not gene editing, but gene therapy, can be used in inhibitor patients, they think, based upon all of the work done in animals that have antibodies. Because when the platelets get to the cell surface, to the wound surface, and release the factor VIII, the inhibitors really don't have time to neutralize the factor VIII, and so it can provide for effective hemostasis. But the other gene therapies, gene editings, really cannot be used in patients with inhibitors. Hi, Johnny. Why does AAV go to the liver? Well, the AAV goes to the liver for two reasons. The first reason is that it's got proteins on its surface, the surface of the viral capsid, that can bind to receptors that are on hepatocytes or the liver cells. But the second reason why so much of it, 90% of what you infuse into a patient goes to the liver. The second reason why so much goes to the liver is because the liver has a unique architect. It has something called sinusoids. What does the liver do? The liver normally, in all of us, filters our blood. And so in order to filter blood, you've got to get the blood into all of the little spaces within the liver can do the filtering. Well, that gets the AAV to those same places, and more of that AAV goes there, which then allows it to continue its journey to get into the hepatocyte. So really two reasons why most of the AAV goes to the liver. Highlights. Has the CRISPR gene editing approach already been used successfully to create therapies in other? Yes, and so uh, we have two approved disease indications, sickle cell anemia and beta thalassemia, where these are also single gene defects. And sickle cell anemia, for instance, is one amino acid defect in the hemoglobin gene. And so two different approaches have been taken. One is to try to fix that hemoglobin gene, change that one damaged amino acid for the authentic amino acid. The other approach is to work with genes that control hemoglobin production. And by doing that, we can destroy a gene that prevents uh, hemoglobin from being made normally in a person, and then that allows a different type of hemoglobin to be made. So these look like they work and work well. They also are having slow uptake. So it's been challenging in this disease area as well. And then, Johnny, please comment on the off-target effect of gene editing using AAV and Lenti. And so with With gene editing, we have so let's speak specifically about gene editing. With gene editing, we have the ability to insert the transgene, whether it's carried by AAV or whether it could be carried by lenti, directly into a region that we want it to go into, a safe harbor region. There may be some off-target effects elsewhere in the genome. We look very carefully for that and can't detect it, but that doesn't mean it's not happening. Just means it's beyond our limit of detection. So as this technology advances and progresses, we need to look very carefully for off-target effects because if it goes somewhere where it shouldn't and it triggers a gene to start making protein, that's the basis of oncogenesis, basically or cancer causing. And so we have to be careful there. But with these current therapies, we have not seen that happen. With the AAV and lentivirus that I described before, where you can get some random integration into the genome with AAV, and with lenti, it randomly does integrate into the genome. We do need to be aware of where it goes, and we need to understand the risk of oncogenesis or cancer-causing events. Not seen though with these current therapies at this point. Upper limit of age for gene therapy. Um, most trials, the upper limit is around 70, just because you start to get into a lot of other diseases. After that, then they can confound the results of the gene therapy. But the case that Brian had asked about in this other disease, transthyretin amyloidosis, this patient was 80 who passed away from the inflamed liver from the lipid nanoparticle and perhaps other comorbidities or cofactors as well. So age is relative, depends upon the clinical trial that one is working on. Prasad, hi. How do we, as a community, encourage pharma to continue to pursue this innovation, given that some have given up on gene therapy? So that's an important point, Prasad. And some of the gene therapies that have not gotten to approval or gotten to approval and haven't sold um, uh, have not had an adequate benefit risk ratio. So we have a whole series of gene therapies that haven't even gotten to phase three. They've stopped after phase one, too. We've gotten some gene therapies to phase three, and then when they compare them to the two gene therapies that are on the market, those gene therapies aren't as effective, and so nobody will buy them. If you have an opportunity to take a gene therapy that will give you an average of, let's say, hypothetically, 50% factor levels, or a gene therapy that gives you an average of 25% factor levels, which one would you take? So I think this is a little black and white here because it's a once and done therapy. So you need to make a decision that's going to give you the best chance for an optimal result. Because unlike taking a factor and then switching to another factor because you think that that one may work better, you can't do that here. Brian, again, looking 10 years ahead, where do you see the genetic therapies for hemophilia and how widespread will their use be globally? Oh, Brian, you always ask those tough questions. I think that we will continue to see penetration of the hemogenics in Roctavian. I think we'll see greater penetration of hemogenics into many more countries than Roctavian, which is currently only in the United States, Germany, and Italy over the coming 10 years as institutions, insurance companies, payers get more comfortable with the concept as more data are accumulated, showing the benefit outweighs the risk. So assuming that that all continues and we don't see some long-term risk that we're not expecting, this will continue. Gene editing has had it's a new therapy. Gene editing really, the form that's being used now was only discovered in 2012, so 13 years ago. So it's pretty that we have products that are on the market for sickle cell disease and beta thalassemia in such a short period of time. Hemophilia is a little more difficult, but we have this first wave of gene editing that's in the clinic or going into the clinic. We'll see how it does and what iterations come along as we go forward. So I would expect to see some gene editing by 2035, but probably behind the gene therapy. The key for the gene editing is not only are you pretty much guaranteed once and done to get an effect, assuming it works, but one can then begin to go into children, which we haven't really been able to do with AAV therapy. Roshni, thank you. Uh, this is a must-watch for all in the hemophilia community. Thank you, Roshni. Is there greater persistence of factor IX due to its migratory processes? So I think what you're referring to is the fact that the active factor not in the base membranes of the blood vessels. It's in the walls of the blood vessels, and that's where it works. So what we measure in the circulation for factor IX is a surrogate for where the factor IX is actually located and where it's working. Um, I don't think that that really has any impact necessarily on gene therapy or gene editing. It's just that we need to recognize that what we're measuring in the plasma is a fraction of the amount of factor IX that is in the body. Can umbilical cord cells be used as a source of stem cells for gene therapy? Um, well, they could be. They could be, but we're using gene editing. Gene therapy is used all autologous cells, so it's using cells from the person. Those cells are removed from the body, they're manipulated, then they're put back into the body. If you do what's called allogeneic transplants, which is what the umbilical cord cells would be, then that gets into questions of graft versus host disease, more morbidity. That's generally done for for cancers as well as for very severe autoimmune diseases. Is there a factor VIII Padua? Well, there is a factor VIII variant that was discovered in Padua, Italy, but it doesn't have the same functions as the factor IX Padua, which gives a big burst of factor IX activity. How could the protein expression of the edited gene be controlled since it's new? Um, so it's controlled by the instructions that are placed with the gene. And so the instructions tell the gene how much protein to make. And that's all worked out in animal models before ever going into humans. And then last question, should gene therapy of hemophilia B be promoted in low-income countries? Yeah, and so that's a really important question, something that certainly has been on my mind for a long, long time. Could this be a solution in low-income countries? And the answer is a qualified yes. Taking factor IX, for instance, we at the WFH, we've been working over the years with St. Jude's to look to develop a trial in some lower income countries to just demonstrate that this is something that could easily be done in a lower income country. And so there will be other forays as well. And some lower income countries have already been in some gene therapy trials. South Africa, Johnny Malangu, who's been on this call, for instance, as well as Margaret Lozello from Brazil, have enrolled a lot of patients in gene therapy. So there's nothing magical about it. The key thing is the price. And gene therapy, by and large, is not expensive to produce. It's certainly more expensive than factor products, but If one could develop public government partnerships, there are avenues to do this. And for instance, the Gates Foundation is pursuing this with sickle cell disease. If we have the will, if we have the advocacy, we can look to move this as it gets more widespread use in high-income countries. We can look to move this into lower-income countries where it would have enormous benefit for patients. So stay tuned, but be thinking about that because all of us are advocates. With that, I'd like to thank you very much for all of your time and attention, and we'll see you next time.